So today my talk will be on single-cell splicing QTLs and their implication in disease and genetic regulation. So this is the okay. So just to set up the background. Okay, this is the point. Great. I was trained as a classic population geneticist during my PhD. So this was, the first successful GWAS back in 2007, and you see that there's a handful of GWAS loci that appeared back then. And then this is in 2018. There was an explosion of GWAS loci, probably 100 times more than when we started in 2007. And what's more interesting is that in 2018, NHGRI stopped making these figures because there's just too many GWAS loci and there's no point making these figures anymore. And we know that most of the GWAS loci are in non-coding regions and only 10% are in coding regions. So we know that even GWAS, we consider GWAS as the core of modern human genetics. There's a broad body of work surrounding GWAS, which is functional genomics, and that's the reason we're here today. So I spent many years to work on this project, which is the genotype tissue expression project. So this project spanned about 12 years with lots of funding, and the goal is to collect a large cohort of people and sample their tissues broadly, right? So the last phase of GTEx is 50 tissues across about 900 individuals. And there's many conclusions from this study. I'll be highlighting one of them, which is that we have an abundance of association between genetic variant and gene expression. So on the x-axis here is the negative log 10 p-value, right? So the lower is less significant, the higher is more significant. And then on the y-axis is the proportion of SNPs. So you see that if we use a nominal cutoff, then almost 100% of SNPs will be associated with expression of at least one gene. But of course, we'll never use nominal cutoffs, right? So if we correct for the number of tissues that we test, still 64% of SNPs will be associated with expression of at least one gene, which means that if we throw a stone at your genome at a variant, then more than half of the chance you will you pick up a variant that is regulatory, right? So we have significant abundance to detect these, genetic versus gene associations. But unfortunately, this insight did not translate into disease. So if we look at the colocalization statistics between tissue-specific QTLs and GWAS, less than half of GWAS loci colocalize with GTEx QTLs. And this is even more dismal for some diseases, right? So for example, for lupus, maybe less than 20%. So in the last few years, the field has moved into single cell and of course, spatial. I don't think the genetic field is spatial yet. Most of them are still doing single cell. And the reason for doing single cell is that we want to sample the correct cell types, right? Because tissue are composed of cells, different cell types, and also correct differentiation stages, and also correct perturbations, right? But my argument today is that in addition of these three, we also have to take a look closely at different ancestry and also different molecular phenotypes. And I'll tell you the reason later. There has been a whole slew of papers published from 2022. So these are the two initial papers, all the way to 2024. And these are really, really high-end papers. But all these papers suffers from the two same problems, right? The first one is that they're all focused on one population, which is European population. And there's good reason for that, right? Because they started this research early, and also these countries are very well funded. And the second reason is that the second problem is that all these papers focus on one molecular phenotype, which is expression, right? But we know that RNA is much more complex than that. There's, I don't know, like 50 different post-transcriptional modifications. One of the most important one is splicing. So that's what we focused on, which is to perform splicing regulation analysis across a large cohort of individuals of non-European populations, right? So so we collected this data set of 1 million PBMCs from around 500 donors of diverse Asian descent. So I work in Singapore, and Singapore has 3 major ethnic groups: the Chinese, which is 70%, Malays, which is 20%, and also Indians, which is about 10%. So here we can classify these cells into different cell types. So most of you would know, there's plasma DCs, dendritic cells, monocytes, natural killer cells, different types of T cells, and different types of B cells. So with that, we could actually look at how much splicing information can we get, right? So I think the reason that people didn't do splicing and people prefer to do expression is because expression is what these assays are designed to do, right? Because they do sided capture. They capture either the 3' end or the 5' end. So the common belief is that we have severe bias towards one of these ends, that we cannot see the full information, which is partially true. But through careful analysis and construction of wet lab and computational pipelines, we're able to show that we can actually get fairly confident splicing statistics from using single-cell data. So this was the previous state of the art. This was 1K1K, published in Science in 2022. So on average, their assay was able to detect about 267 genes per cell. 267 alternatively spliced genes per cell. Sorry, I should. But we were able to drag the statistics up from 267 to be somewhere between 1,000 to 2,000, depending on the cell type, right? So about 4 to 5-fold improvement. And some of you might be asking, are we just sequencing more deeply, right? If we're sequencing more deeply, of course, yeah, we get more expressed genes and, of course, more alternatively spliced genes. But that's not the case. So here on the x-axis is the number of expressed genes. On the y-axis is the number of alternatively spliced genes. You see that there is a linear correlation as we intuitively would imagine. But wherever you draw the vertical line, our data set is always about 4 to 5 times more than the 1K data set, right? So, and with this, now between 1,000 to 2,000 alternative spliced genes, we can talk about genome level, genome-wide level differential splicing analysis. So here is what we did is that we stratified the individuals into different ethnic groups: Chinese versus Indian, Chinese versus Malays, and Indian versus Malays, and we detected genome-wide differential splicing patterns, right? So here I want to stress one thing. All these individuals, all these 500 individuals are healthy individuals. So what we're doing is we're comparing healthy individuals against other healthy individuals, right? We're not comparing healthy individuals versus diseases. So, and the question now is like, why would there be differential splicing between 2 healthy individuals? We expect their livers to function similarly, their hearts to function similarly. Why would they have differential splicing? And then it turns out that this differential splicing can be harmless, right? So here's one example we found is that there's this one isoform for this gene called SPSB2, and there' a variant RS110 that disrupts this splice site, right, from a G to an A. And then this 5' splice site is forced then to use a downstream cryptic splice site that's 12 pp downstream. So basically you get 4 amino acids less than what you would have gotten using if there was no disruptive variant, right? But 4 amino acids might not be a huge deal in terms of protein folding. So here this variant has a allele frequency gradient from Chinese to Malays to Indians, and we see that the usage of this canonical splice site corresponds exactly to the allele frequency differences, right? So it's the allele frequency differences driving the differential splicing. And then we combine with the genetic data, we can actually map splicing QTLs. So we mapped about 11,000 cis sQTLs and then 607 trans sQTLs. I'm going to tell you two stories because I think humans think better in stories than numbers. So the first story is that we were able to infer a regulatory pathway using the single-cell data set alone. So here, first we identified two cis regulatory SNPs regulating the expression of H and RMP-LL. And these two regulatory SNPs are functioning through different cell types, right? One is functioning through naive T cells, and one is functioning through memory T cells. So you can see these two SNPs, their fine mapping statistics, they're fairly independent. They're 36 kb apart, and also their R squared is less than 0.3. So we're fairly confident that they're independent. What's more interesting is that these exact two regulatory SNPs and the exact two same cell types shows a regulatory relationship, a trans-regulatory relationship to this gene called PDPRC, which is also called CD45, right? So what happens is that these two SNPs would push or cause a switch from the RO isoform to the RA isoform, right? So exclusion or inclusion of exon 4, 5, and 6. And we know, and we saw a very strong colocalization pattern between the eQTL effect, cis eQTL effect, and the trans sQTL effect. So you see that whenever you have a cis eQTL effect, you have a strong trans sQTL effect. And what happens is that this gene, HNRMP-LL, is an RNA binding, produces an RNA binding protein, right? So it produces an RNA binding protein, comes and searches for exon 4, 5, and 6, and marks it for splicing. Right, so that's what happens, and we can, because we have single-cell data, we can stratify these cells into two subgroups, whether they have expressed HNRMP-LL or not expressed, and you see a big difference between the splicing pattern of PDPRC. The last one I want to show you, this conclusion might be a little far-fetched, is that some of you would recognize CD45 as a key marker for T-cell differentiation. Right, so it's a key marker between naive versus memory T cells. So then we ask the question whether these two variants, because they change the splicing pattern of PDPRC, can we use Mendelian randomization to show that they actually change the T cell proportions? And that's exactly what we see. Right, there's a very strong correlation between the cis effect and the trans effect on the cell type proportions. Okay, so I guess just to quickly summarize that last slide, we were able to infer this whole pathway from the genetic variant to cis regulatory effect to trans regulatory effect and to cell type specific proportions using just a single cell data set alone. And we also connect our cis QTL regulatory statistics to disease. So that's what we did. So here I'll explain the figures first. On the x-axis is the different cell types. On the y-axis is the different genes, and the color codes for different disease types, right? So for example, for this one, TCHP, we saw a strong signal for Graves' disease, right? For Graves' disease, and in these cell types, in monocytes, in natural killer cells, and in T cells. So here we see that there's a strong colocalization between the sQTL statistics and Graves' disease GWAS. But more interestingly, we also observed that whenever we have an sQTL, there is an eQTL statistics. Yeah, so see this is eQTL, and this is sQTL. So that's interesting to us, right? So now the question is, does the sQTL lead to the eQTL, or does the eQTL lead to the sQTL? And I'm going to state the conclusion first. Our conclusion is that the sQTL leads to the eQTL, and I'm going to show you the evidence later. So what's more importantly is that this colocalization is cell type specific, right? So it shows up in monocytes, in natural killer cells, T cells, but then disappears in B cells, and also disappears in all these other cell types, right? So if we had looked in this in GTEx, we wouldn't be able to find this colocalization because 2/3 of the cell types don't have this regulatory relationship. we were able to functionally validate that, but before I show you the experimental data, I'll show you the gene structure. So this variant RI744 is sitting right on top of the splice site of exon 4 of TCHP. And more importantly, remember I mentioned that I want to make the argument that ancestry specificity is important, and here's one of the examples. So because RI744 only appears in East Asian populations and Southeast Asian populations, if we looked at European populations, we wouldn't be able to find this signal, right? So, so now answering the question whether the eQTL leads to the sQTL or sQTL leads to the eQTL, so we were able to clone this TCHP exon 4 out, and it's surrounding 200 BP introns into a mini gene construct. And this is done by Donald and Xavier, a very talented collaborator. And so the G is the risk variant, and then the A is the non-risk variant. So you see that the G variant leads to, the other way. G is the non-risk variant, A is the risk variant. So the non-risk variant leads to normal splicing, whereas the risk variant leads to intron retention, right? So you have intron You still have intron retention, and then that leads to nonsense mediated decay, which leads to decreased expression, which leads to eQTL. So conclusions: ancestry bias alternative splicing is widespread, and some of them can be explained by minor allele frequency differences. And also trans-splicing effects can be mediated by cis-eQTL effects on RNA binding proteins. I showed you one pathway from 2 independent SNPs to H&RMP-LL to CD45 to cell type proportions. And last but not least, TCHP is a putative causal gene for Graves' disease, and also its risk allele only appears in East Asian populations. So I'm going to skip these speculative slides, and I want to highlight a few people who are the heroes behind the curtains. So these are the 3 co-first authors of this paper, and also these are the people who collect the cohort and generate the data, right? Ken Holm, Sham, Jay, and Mo Young.